My lab is in the Department for Biochemistry and Molecular Biology, as you said, and I'm actually in the Center for Brain Health on campus. And so this is actually a picture of my lab. So I run a basic biology wet lab. These are my students. I have some grad students, some undergrads, and this is actually in front of our building, which if you've ever walked around on campus, we're right next to the corner hospital, and we're the giant building with what looks like starfish on the front, but they're actually brain cells. So if you want to come over and see some brain cells on the front of a building, we've got the only one on campus that I've seen. So my lab focuses on trying to understand how genes are regulated in the brain, and specifically in the context of brain development and behavior. And this is actually a picture of a painting that's hanging in my lab that my sister did when I opened my lab in 2019. That is sort of an artistic interpretation of looking at how the interactions between brain development and brain cells can be controlled at the level of our genes. And so why should I care about genes, and what are those? So you may have to remember back to 10th grade biology, but in a nutshell, genes are the blueprints for who you are. So they're encoded within our DNA, within every cell in our body, and they contain all of the information that it takes to encode you. And so your genes have the ability to code for your skin, your brain, your feet, and everything in between. And so this really is the sort of map for making all of us. But most of you are probably hopefully aware, we are very much more than the sum of our genes. You can't sequence someone's genome and know every single thing about them. There is no gene that determines what your career path will be, whether you become a chef or a teacher or a nurse, or whatever you choose to be. That is not predestined in your genetic code. And so your genes and your environment is really what forms who you are. So you are a product both of your environment across development, the choices you make across your life and your lifestyle, and of course, these things interact. And it's really the interaction between your genetic code and your environment that forms the basis for who you are as a person. And so sort of at its heart, we're interested in trying to understand what are the mechanisms that allow this to happen? How do our genes and our genetic code that we're born with dictate some of the choices we make about our lifestyle, or what type of environment we choose to enjoy? And the converse, how does your environment or your experience across your life influence how your genes are used? And of course, we're interested in this not only because it's fundamental for how we are shaped as human beings, but these processes are also shown to go awry in diseases, especially in the cases of neurodevelopmental or neuropsychiatric diseases, where we really understand very little about what the disease is caused by and what we could potentially do to treat it. And so my lab has focused in primarily on this interaction between your environment or your experience and how your genes are used. And the mechanism, or one of the main mechanisms for how this happens, is a field of study called epigenetics. And this has been popularized in the press over the last couple of decades. But what this means, so it's epigenetics, and the Latin translation means on top of genetics. And so it's the way that basically everything in your environment can influence how your genetics are used. And this is the main mechanism for which environmental control can dictate changes in gene expression. And so my lab is interested in trying to understand how epigenetics occur in the brain in the context of development and also in the context of disease. So this is another painting from my office, which was done by a young woman with autism. And so one of the candidates for some of the underlying etiology of autism is changes in how the epigenetic machinery is influencing brain gene expression and development. And so how do we actually go in and study this in the lab? Well, my lab uses mice to try to understand the relationship between the brain and genes and behavior. We can use mice because they show many of the behaviors that we're interested in studying in the context of brain diseases. So for example, this is an example of a social interaction test in which we're looking at how the test mouse, which is outside of the mouse jail, interacts with the mouse inside the mouse jail. The mice actually don't mind being in jail. It's not that big of a deal. But we can look at how they interact and draw some conclusions about disorders in which social interactions are impaired. Now, obviously, mice are not tiny humans. This is the human and mouse brain to scale. However, many of the underlying building blocks of the human brain are well conserved in the mouse. So they have many of the same regions of the brain, they have the same fundamental cell types, and importantly for us, their genes are regulated in basically the same fashion. So this is a picture of a couple of different types of brain cells that we can grow in culture, and they look very similar between mice and humans. And so the real strength of using these model systems is that we can go back and forth between brain samples and cells, brain samples and behavior in a way that's really not possible working directly with human subjects. And so I'm going to give you a little vignette about some work that we are doing in the brain. And I'm specifically going to talk about how we're looking at how genes are regulated in microglia in the brain. And so what are microglia? They are the immune cells that live in your brain. So we have many different types of immune cells in our body, and the brain has its own immune cells, and they're the microglia. And what's shown here is a picture of a part of a brain, and all of the little golden stars are microglia. And so they are spread out across the brain, and they are constantly surveilling the brain, looking for disease and infection. So they make up about 10% of the total brain cells, and they're incredibly important for brain injury and disease. And they do actually a whole bunch of other functions that are really important for brain development. And so microglia can shape how connections form in the brain. And so in early development, we actually make a whole bunch of brain cells that form connections. And these connections actually are produced in abundance. So they have an overconnection. And what we want to do to correctly have different brain regions talking to each other, we need to shape those connections through experience. And microglia can help do that. They can help trim away the inappropriate connections, leaving only the proper connections that are important for the brain to function. And so my lab is interested in trying to understand abnormal microglia and their role in potentially shaping an abnormal brain. So microglia do a bunch of different processes. They can help brain cells connect. They can control the number of brain cells. They can control brain signals and response to the brain environment. And all of this is potentially disrupted in autism, but not unique to autism. Actually, most brain disorders show some kind of disruption in microglia function. And so we wanted to understand how do microglia regulate their gene expression. And we do this in the lab by treating them with a bacteria product, so LPS, which is part of the outside of a gram-negative bacteria. And this turns the homeostatic microglia, when you add the LPS, into activated microglia. And you can sort of think of this as angry microglia. They're responding to an inflammatory event. And what we've done is we've showed that microglia will transition to these angry sort of phenotypes where they're producing a bunch of inflammation molecules in the brain when you give repeated doses of LPS. And this enhancement, when you give 2 doses, is what we've termed priming. But if you give them repeated doses up to 3 or 4 stimulating events, this priming event goes away and you actually return them to their sort of happy baseline state. And we really don't understand how this happens. And so we're working on the hypothesis that there's some sort of epigenetic switch between priming and tolerance. And if we could understand how this works, we might be able to design new treatments to mitigate neural inflammation and disease. And so we've started looking at what the morphology of the microglia do in the brain. So they go from being sort of these homeostatic and branched microglia to sort of rounding up and sending out a whole bunch of processes when we start to give them more LPS. This is just an example of one of the types of experiments that we're looking at. And we're trying to understand how long lasting these changes are, how they relate to brain function, and ultimately behavior. And so I will just give you a couple of conclusions. If you take away nothing else from this talk, epigenetics is awesome. So are microglia. And I'm really excited to be part of this program and to get to know all of you and meet you over the next several months. And so I will end with that.
Your timekeeping is awesome. And I think you're perhaps just edging ahead in the competition for the coolest slide deck. At least you pushed the standard pretty high. We might have a vote on that at the end of the series. There was some joking in the group before we let everybody in about whether they should exchange slide decks and talk to each other's slides. Wow, we've got time for some real Q&A. I think Megan's up first for this one.
Yeah, thanks for that helpful explanation. I found the people in biology in those types of fields have the best. They actually have like a website that I was given where they can go and get the coolest little infographics and stuff. I don't know if you know what it is, but I just wanted to know, what is the LPS like? What is that like replicating in real life though that would affect a microglia? But,
yeah, so I kind of glossed over that. So LPS comes from the outside part of a gram-negative bacteria. And the most common example is like E. coli. So we actually inject the LPS into the periphery of the animal to mimic like gut infection of E. coli. So it's probably equivalent to sort of the kinds of symptoms that you get with like a food poisoning or something like that. So we look at sickness behavior in the animals. So they stop eating, they lose weight a little bit, they kind of hunch up and stop grooming themselves. They just look unhappy. But it only lasts for about 24 hours, so it's pretty mild. So it's sort of like having a day or two where you're maybe sitting at home in bed.
And just to follow up, the gut health has been linked to autism, though, these kind of and other problems, right? So there's some reason for doing that to them. Okay.
Yes, yeah. So we use it as a convenient way in the lab to mimic an infection. I have a whole branch of my lab that works on microbiome brain interactions and looking at how changes in the gut microbiome with gut inflammation. We have a mouse model of like colitis. How that can influence brain development and microglia. So we're looking for the signals that the gut can release that talk to the microglia in the brain.
Meanwhile, Lydia in the chat is already doing the leading scholars thing and drawing you into a wider conversation here. Annie, do you just want to take a glimpse at the chat and put a bookmark there for everyone?
Yes, interesting. Yeah, so the people can read it, but sort of the general questions about race in medicine and science, that is a whole giant topic and can of worms. Yeah, and one of my other hats that I actually wear for my department is I'm the chair of our EDI committee. So this is something that we think about, not just in the impacts on science, but who's doing the science and in what.